Okay, in this video what we're going to do is we're going to do the functional anatomy, and I'm going to talk about vision and the visual deficits. So we're going to have a chance now to look at some of the structures that we've reviewed, and we'll again talk about these visual deficits that occur. So the visual pathways within the retina, we have rods and cones, and the role here today is not to look at the photoreception. So we're not going to look at the receptors themselves, but suffice to say that light hits the retina. Basically, we have action potentials that fire in the rods and the cones. Now, those rods and cones will synapse onto bipolar cells, which then synapse onto the ganglion cells. And the ganglion cells exit the eye to form the optic nerve. So here we have the retina. So here's the retina with those photoreceptors. The photoreceptors ultimately connect to ganglion cells, and the ganglion cells, which I'll draw now in green, now exit and form the optic nerve. So they basically now receive the signal and they enter the central nervous system. Interesting thing about the optic nerve, within the eye itself, it's unmyelinated because the myelin would interfere or would reflect any light reaching the retina. So again, the myelin is white and that would be reflective, whereas the optic nerve only becomes myelinated once it actually exits the eye. So this is where I'm drawing here. The thicker is where it becomes myelinated. So again, so that's the signal, and again, the action potential now is sent towards the central nervous system from those receptors. So the visual pathways here, we've seen this diagram in your anatomy. Now we're going to talk about, here's the, we're looking at from below, so this would be the right eye. Here we have the left eye. Those signals, those would connect here to structures that, and if we talk about the different structures, we know that this is the optic nerve, or cranial nerve 2. So the optic nerve, we get to an area here in the middle, which is the optic chiasm. And then beyond the optic chiasm, which I'll draw here in blue, both on these pathways here, is what's the optic tract. So we have the optic nerve from the left and the right eye joined form the optic chiasm. And then we have the optic tract with the information that travels towards the central nervous system at this point. Again, everything we've seen so far, we're in the periphery. So now, as we continue with the visual pathways, axons from the retinal ganglion cells relay visual signals as action potentials via the optic nerves and optic tracts, now that's what we saw in the previous slide, to the midbrain diencephalon junction. And these axons basically go to one of three sites. We have those going to the lateral geniculate body. We have some of those axons terminating in the superior colliculus or superior colliculi, and then we have others going to the pretectal nucleus. So that's the where these visual pathways actually terminate. The lateral geniculate nucleus, the lateral geniculate nucleus, the superior colliculus, or the pretectal nucleus. Now let me just talk about what these different pathways do. Those signals that reach the lateral geniculate nucleus, and we know that now we're talking about the visual pathways, and again we said lateral for light, L for light, or lateral geniculate nucleus, and this is the main subcortical region that processes visual information for perception. So the majority of the retinal axons terminate in the lateral geniculate nucleus. So this is where most of them go. And ultimately then from the lateral geniculate nucleus, the signals then are sent to the principal or the primary visual cortex, which you know is the surfaces of the occipital lobe, and especially the banks of the calcarine sulcus is where those fibers would terminate. Again, they go from the lateral geniculate nucleus, and then from there we would have fibers that would go to the occipital cortex. Now, besides the lateral geniculate nucleus, we said we had some fibers that terminate at the superior colliculus. And those fibers that reach the superior colliculus basically control saccadic or fast-moving eye movements. The superior colliculus coordinates visual, somatic, and auditory information, adjusting movements of the head and the eyes towards a stimulus. So again, if you're walking somewhere and all of a sudden a friend who's, let's say, to your left in the parking lot yells out your name, you're going to now direct your visual attention to where the sound came from. That's those fibers going to the superior colliculus that allows you to do that. And then we also have some fibers that go to the pretectal area. The pretectal area, again, remember the tectum was the area of the midbrain that was distal, or posterior to the cerebral aqueduct. The pretectal area, as we're going to see a little later on in more detail, controls pupillary reflexes, and especially the pupillary light reflex and the accommodation reflex. So the fibers that actually go to the pretectal area basically are involved in the pupillary light reflex and the accommodation reflex. And now we can look at some of these structures that we've seen before. So again, here we're talking about a transverse section of the midbrain diencephalon junction. Hopefully you remember this. If we look here, we have the pulvinar. So we know the pulvinar is part of the diencephalon. We have the pineal body, again, part of the diencephalon. We're looking at structures here. Now, part of the midbrain structures, we have the superior colliculus here, the superior colliculus here. In blue now, I'm going to an area that is known as the pretectal area. So the pretectal area, as I'm showing here in blue, we have the cerebral aqueduct. So we know, and if you look at the shape of the cerebral aqueduct, you know you're just at an intersection between the third ventricle and the start of the cerebral aqueduct. Other structures that are important here, again, this very dark fiber here is the optic tracts. And if we follow the optic tracts, we can see that the optic tract goes to this structure here, which is the lateral geniculate nucleus. So we can see this fiber running to the lateral geniculate nucleus. From the lateral geniculate nucleus, we're going to have some fibers that are going to go towards the V1 or the primary visual cortex. But we also said earlier that we have some of these fibers that will go to the superior colliculus and some fibers that will project to the pretectal nucleus. So again, that's where we have the optic tract where the fibers will terminate. Again, most of them, though, do go to the LGN. So the primary visual pathway, let's look at now how light is actually processed. And what I have here is what's known as the retinogeniculocalcarine tract. So let's look at what we're talking about here. We're talking about the retina, so that's where these fibers originate, from the retina. The geniculo, we're basically talking about the lateral geniculate tract in the thalamus, and calcarine is where they actually end. So this is the visual pathway from the retina all the way to the primary visual cortex. And again, this is where we have our perception of light. So let's just go over some structures here in this diagram. We're looking from above. So here we have our left eye, the right eye. And what's shown in this circle is the visual field. This is what you actually see. So these are the visual fields. And if you look, we have the visual field of the left eye. And in green now, I'm going to draw the left visual field of the left eye. And in red, I'm going to draw the right visual field of the left eye. Now, if we do similar in the right eye, we have the left visual field of the right eye. This is what you're actually looking at. And then in black now, I'm going to put the right visual field of the right eye. So the way light travels, so let's go now and see the actual processing of that information. So light travels in a straight line, so the visual information, so from the left visual field of the left eye, the light will actually hit, so if this is my nose here, So this is the person's nose. So this is known as the nasal retina. And if I follow the signals now from the nasal retina in green, I'm drawing the pathway now, it crosses over, it goes to, this here is the lateral geniculate nucleus, and from there it now synapses onto what are known as optic radiations, and now they go towards our signal, which is the primary visual cortex on the right side of the brain. So that's the processing of the information from the left visual field of the left eye. Let's see what's happening now, or how the information is processed from the right visual field of the right eye, that area that I highlighted in red. Well, this information now reaches what's known as the temporal, because the temporal area is on this side of the retina. So your temporal retina receives the light from that visual field. The signal is carried in the optic nerve, now in the optic tract, and it goes to the lateral geniculate nucleus on the left-hand side, and now through what are known as the optic radiations, these signals now go towards V1, or the primary visual cortex, which is again the banks of the calcarine sulcus on the left side. So that is the information that is flowed from the left eye, from the left and the right visual fields of the left eye. Now let's do similar pathways now for the right eye. We saw that the left visual field of the right eye is this area that I drew in blue. That light now will hit the temporal retina on the right. And now if I draw the pathway, this is the pathway here. It now projects to the lateral geniculate body on the right. And now through the optic radiations, they now project and terminate or end. Those axons would project to the primary visual cortex on the right-hand side. And if we now look at the right visual field of the right eye, that area that I basically drew here in black, now again light would reach the nasal retina of the right eye. We have now the signals coming in through the optic nerve, crosses the optic chiasm to the area, and eventually goes to the lateral geniculate nucleus on the left side, and those fibers now will project all the way to the primary visual cortex, V1, on the left-hand side. So what can we remark? We notice now that in the area here in the left occipital cortex, so here we're talking about the left occipital cortex, this area here receives the information from the right visual field of the left and the right eye, versus the right occipital cortex receives visual information from the left visual fields of the left and the right eye. So that's how the information flows through the reticulo-geniculocalcarine sulcus. So again, these are the pathways that the light would take. Now here's sort of a description or a diagram showing you these pathways where they've actually dissected parts of the occipital cortex and part of the temporal cortex so you can see these actual pathways. So again, if I draw, I would have my, I'll use red marker here. So here's my right eye, here's my left eye. Again, we're looking from an inferior aspect. We have the connections here that have been cut, but here we would have the optic nerve or cranial nerve, the optic nerve or cranial nerve 2. We have the optic chiasm here with the signals. We have here is the optic tract, and this area here is the lateral geniculate nucleus. And now you can see these optic radiations. Now we haven't seen those in class. Now you can see those fibers end at the occipital cortex, and again the banks of the visual cortex, an area that we refer to as the primary visual cortex or V1. So again, you can see if our cross section here, we're an area really close to the midbrain. Here you have your cerebral aqueduct, And you can see the other structures. Here we had our mammillary bodies, just as a reference point to where you're located here, where this cross-section takes place. But this is just a beautiful dissection which allows you to see the optic radiations. So these here are the optic radiations shown here, both on the left and the right-hand side. Or if you look at these blue arrows that they have in the diagram, really showing the optic radiation. So it's just a beautiful dissection to allow you to see that reticulocalcarine tract in its entirety from, again, at least I drew in the left and the right eyes, but you can now follow it and see where those pathways would go. So let's talk about damage now to the reticulocalcarine sulcus. What I'm going to do, and I'm going to redraw these pathways that I drew earlier, just so we can look and see what these different lesions would occur. And here what I'm going to have is, we're going to talk about the left eye, and here's the right eye. And again, remember, this is what we're actually looking at. So I'm just drawing midway so we can basically draw sort of what we're seeing. So this is, again, the left eye versus the right eye. I'm going to now draw in red. We'll talk about the, again, so here we have left eye, right eye. We're looking from a superior aspect in this diagram. So here we have the left visual field of the left eye. Again, it goes on to the nasal retina, travels in the optic nerve. It will cross the optic tract, go to the lateral geniculate nucleus, and from there it will synapse onto other neurons, which now travel and basically end in at V1. We now draw as far as what from the left eye to right visual field. Here we have the pathway. Now it basically remains ipsilateral. It goes to the LGN on the left-hand side. It goes to the optic radiations, and it terminates at V1 on the left-hand side. Now we focus now on the right eye. Basically, again, in the left visual field of the right eye in blue. So this is what we're seeing. We have the light hitting the temporal retina. Information now doesn't cross through the midline, remains ipsilateral. And now we have from the LGN to the V1 on the right-hand side. And then finally we have the visual field, the right visual field of the right eye. And the signal transduction crosses the midline, travels in the optic tract on the left, goes to the lateral geniculate nucleus on the left-hand side, and now ends and terminates in V1 on the right-hand side. So if we look now at lesion 1. So lesion 1 here is where there's an actual disruption in this diagram here in the optic nerve. There's a lesion. And what happens, we lose, again, if we talk about our red, green, blue, and black, if we're looking at what we're seeing here, you can see that we lose the green. So we would lose green, and we would lose the red mark, or basically what happens now is if we lose this, is you basically have loss of vision in your left eye. The right eye would remain normal. It's unaffected in this type of lesion. Signal occurring at 2. So here we have a lesion where we have the optic chiasm, where there's a lesion of the optic chiasm at 2. And so what we're losing here, if we look at the fibers that actually cross the midline, we can see we're losing the red. So here I'm just going to look at the eye. So we're losing this area here, this visual field. And we're losing the black, which was this here. And so this person then would lose sight in the left visual field of the left eye and the right visual field of the right eye, with a lesion occurring at the optic chiasm as diagrammed here in the picture. Number 3, lesion 3 now is where we have a lesion affecting the optic tract. And if we look at the optic tract, what we're losing the green. The green area here was our right visual field. So we're basically losing the right visual field of the left eye, and we're losing the right visual field of the right eye. So this would be the area here shown in black where there would be loss of vision. So this again is what you're seeing, so this would be our loss of vision. If we had a lesion at 4, and this is a complete lesion here of the optic radiations, and again, if we have an area where the optic radiations on the left side of the body were interrupted, that information is interrupted, again, we would lose the right visual field of the left eye, and we would also lose the right visual field of the right and left eye. So we're losing the right visual fields of both of these eyes shown here in black. So we would lose, again, the green and the black. And then finally, if we had a lesion here, lesion 5 is in the primary visual cortex. And here is we would have a lesion where we would see something what's known as macular sparing. So you don't lose the entire visual field on the right. There's this little area where there's this macular sparing occurring, where that area supplies by the macula, because of the number of receptors in the macula, would be intact. So those would be the primary lesions you would see, the visual deficits you would see with these types of lesions. You can go online, or even your textbook has some nice examples of some of these visual deficits. So visual deficits, so how do we sort of have different terms for these visual deficits? Visual deficits are generally described by the visual field, so it's what your deficit is, so the visual field losses, rather than by the anatomical structures injured or damaged. The anopsia refers to a loss of vision. So hemianopsia or a quadranopsia are losses of vision in one hemifield or in one quadrant of the visual field respectively. So again, you have hemi, which is where you're losing half of your visual field, or a quadrant where you're losing less than half of a given hemifield. And then another term we can use is either hetero or homonymous anopsias. These indicate visual field losses and which are not on the same in both eyes. So a heteronomous indicates a visual field losses which are not in the same in both eyes, whereas a homonymous indicates a visual deficit that is similar in both eyes. So if we go back now to the example that we had on the 2 pages ago where we had our clinical deficits, and I'll just redraw these deficits. Total blindness of the left eye is where we had a lesion 1, and that was the total blindness here is what would be referred to as total blindness of the left eye. Again, here's our left eye, here's our right eye. That's what we're actually seeing. So drawn here in black is total blindness of the left eye. Our second lesion, where we had a lesion affecting the optic chiasm, is known as a bitemporal heteronomous hemianopsia. And if I basically draw sort of the line here, this is where we had this loss here in the left visual field of the left eye and the right visual field of the right eye. So it's bitemporal, so it's both sides of the temporal visual field. It's heteronopsia because, again, these are different. The left eye has a left loss versus the right eye has a right visual loss. And it's hemianopsia because, again, it's half of our visual field. Area 3, the right hemianopsia, is the lesion we had at 3. And this is here where we had visual losses of our right visual field of both the left and the right eye. So it was a right homonopneus because it was, again, the same visual field on the right, and it was a hemianopsia because we're losing the entire half of the visual field. That was a lesion that we had in the optic tract on the left side. Number 4 now is the right homonymous hemianopsia, is where we had a lesion affecting the optic radiations, the complete optic radiations on the left side of the body, and where we have this complete loss here. And again, this is where we have a lesion of the complete optic radiations on the left side of the body. And then finally, where we had the lesion affecting in the visual cortex on the left-hand side is we would have this visual deficit here. And so this is the visual field that you would lose. So you can see this visual field. So now this would be a right hymenopsia. But if you notice that there's macular sparing, because in this area supplied by the macula, there is sparing, or there is still vision in this area supplied, or where the fibers originate from the macula. So that's how you can differentiate a lesion in V1, so the primary visual cortex, because you have this macular sparing, versus a lesion either in 3 or 4. We're losing the right visual fields in both of these because the lesions are occurring on the left side of the body, the left either optic tract or the left optic radiations. So again, remember, so everything, the information from the left visual fields ends up in the right cortex, and the right visual fields from both eyes end in the left cortex. So that's how we would name those different deficits that we have seen earlier. Now, one additional thing I'd like to talk about is the visual reflexes. And here we're talking about the pupillary light reflex, and this is where we have light reaching the retina, and you know that in response to bright light, the pupils will constrict. So now we're going to talk about the pathways for the visual reflexes. Where here, like any other reflex of the body, you have an afferent limb and you have an efferent limb. So the afferent limb is sensory information that enters the central nervous system, and in response to that sensory information, you have a response that takes place, the efferent limb. So if we look at the visual reflexes, and here specifically the pupillary light reflex, let me talk about the different afferent and efferent information. Well, the afferent fibers, the information carried into the central nervous system, is carried by, in red now, I'm going to redraw this in red, is the information that enters through the optic nerve. So the information starts at the retina. We have the optic nerve, the optic radiations, and now we have a signal that is actually sent to what's known as the pretectal nucleus. We will see this shortly. Pretectal nucleus is in the midbrain area. Again, remember what we referred to as the tectum is the area that lies posterior to the cerebral aqueduct. So the pretectal nucleus we saw earlier. So that's the information carrying into the central nervous system, the light information. Now, I'll also draw the information coming as you would, since we're talking about information here in the left eye, the right eye, information, again, I'll just use this in red, through the optic radiations. It now reaches here. We'll go to the pretectal nucleus on the right side. So that's the information that would be light, that would be actually shone into the left eye. Now, how about the efferent limb of that reflex? The efferent limb is from the Edinger-Westphal nucleus. Now, we saw the Edinger-Westphal nucleus here. So it's the accessory oculomotor nucleus, the Edinger-Westphal nucleus, and you know that we saw that, and we'll see a picture shortly. So from the pretectal nuclei, we have fibers, and I'll draw these in green here, that project to the pretectal nucleus on the left and the right-hand side. And in response to that signal, the pretectal nucleus is actually part of the parasympathetic system, which regulates the diameter of the pupil. And now we'll basically talk about the efferent portion of the limb. So from the pretectal nucleus, carried along the oculomotor nerve, cranial nerve 3. So here we have cranial nerve 3, which carries the efferent signal, which now goes and activates the sphincter muscle. So that's the overall pathway, the afferent and the efferent portion of the reflex. Now I'm just going to erase everything here real quick so we can now use this diagram in a little more detail to see what happens when we basically will shine light in the eye. So now we can start back with this diagram here so you're aware of all of the different structures. And now let's talk about where we take a flashlight, the pen light, and you shine it now into this example here in the left eye. So now I have a flashlight and I shine it into the left eye. So what is the response we get? So let's think about the pathways now. So the light will hit the retina in the left eye. We have the afferent portion carrying the information to the pretectal nucleus. And again, I'm going to draw from the, we're talking about light reaching both the retina on the nasal and the temporal side. So this information here travels to the pretectal nucleus both on the left and the right-hand side. Now from the pretectal nucleus, we have activation. So now there's bright light from both the left and the right-hand side. And now we have activation of the Edinger-Westphal nucleus. And in response, the Edinger-Westphal nucleus, both on the left and the right side of the body, sends signals to the sphincter pupillary muscle. They increase their activation, and the muscle constricts, and we have pupillary constriction. So from shining light in the eye, the pupil on the left eye will constrict. So we get constriction. So if we shine light in the left eye, the left pupil constricts, and that's known as the direct response. We shine light in the left eye, the left pupil will constrict. But because of this bilateral nature of the reflex, shining light in the left eye also leads to constriction in the right eye. So the right eye will also constrict. We just drew the pathways. You can see that there's the overlapping of the signal, and that's known as the indirect response, or the consensual light response. So the consensual light response refers to light being shone in this example in the left eye, and the contralateral eye not receiving the light also constricts. That's known as the consensual light reflex. So you can see again that there's either direct or indirect pathways. Now, if we think of the use of this reflex, I mean, we often see in different examples. So let's say you saw a person who was unconscious. So what is sort of a paramedic would do? The first thing that the paramedic would do would be they would get to the site. First thing is, is a person breathing? So you check their breathing. Second item that the individual would do is, do they have a pulse? So they're breathing, yes. Do they have a pulse? Yes. And then you see them often take out a flashlight or like a pen light, shine it into the left eye, shine it into the right eye, and what they're looking for is a response. And the reason that they do this is what they're looking for is the integrity of the brainstem. Because if there's a major lesion affecting the brainstem, you lose the direct or the consensual or both of these responses. So again, it's a way for the paramedic or for a physician to actually assess the integrity of the brainstem. Again, the brainstem, it's important for these light reflexes, but it's also important areas regarding like your breathing, and a lot of autonomic function occurs in the brainstem area. So again, it's a quick indicator of how well the brainstem is operating. So here, if we look at our cross-section of our midbrain that you've seen in class, so let's look at some of these structures. Here again, we have our pulvinar complex here. Here's our cerebral aqueduct. Here's our superior colliculus. Here's the superior colliculus here. Here we have the area in red that I'm going to draw right here is the Edinger-Westphal nucleus. And I'm going to just now draw the entire in green on the left and the right side is the oculomotor nucleus. So you can see the Edinger-Westphal nucleus, and in green here you can see some of those fibers which are part of the cranial nerve 3. So the oculomotor fibers you can see here emerging on the anterior aspect between the two. Again, here's our crus cerebri. And you can see here midline these fibers which are part of cranial nerve 3, which again would carry the efferent portion of the pupillary light reflex. So visual deficits in the reflex activity, again, you can redraw these pathways if you'd like, but the optic nerve lesion, so if we had a lesion of the optic nerve, cranial nerve 2, so if we had a lesion affecting here, would interrupt the afferent limb of the reflex and abolishes both the direct and the consensual, that should be consensual, responses in the blind eye. So again, if you had a lesion here, you would shine light in your left eye in this example. So neither the left nor the right eye in this type of lesion here would respond. So you'd have no direct and no consensual light response. However, both pupils would react when the good eye is stimulated. So this example here is if I take the light now and I shine the light in the right eye, now both the left and the right eye would respond to that light that I would be shining into the right eye. So again, even though we would have this deficit of the optic nerve on the left-hand side, both the left and the right eye would respond to direct light being shone into the right eye. And then here now at the bottom, we have an oculomotor nerve lesion. So now we're talking about in blue here, affecting the oculomotor nerve, cranial nerve 3, interrupts the efferent limb of the reflex. And here it would result in a pupillary dilation and loss of both the direct and the consensual responses in the ipsilateral eye. And what we mean by this is if we had a lesion of cranial nerve 3 on the left-hand side, the left eye would not have a direct response, meaning if you shine light in the left eye, it would not constrict, and it would also lose its consensual response, meaning if you shine light in the right eye, the left eye would not constrict because of a lesion here in the oculomotor nerve. So again, the example here where we have the oculomotor nerve, we would get pupillary dilation because you would get sort of paralysis of the sphincter. The constrictor muscle would be paralyzed, so you get dilation. That's also known as mydriasis. That's the term we use, mydriasis. And again, you would lose both the direct and the consensual response in the ipsilateral eye. And that's the end of this visual video. So again, review the different pathways, and it just gives you an idea of some of the structures we've seen and how they lead to clinical deficits when we have lesions in these different areas.